Good morning, everybody, and thank you very much for joining our SPHN webinar. We are very happy about the numerous participation, which is probably due to the interesting topic, but also probably due to our guest speaker today. He's a widely known expert working on the interoperability pulse of time, so to say. I'm very grateful that he accepted our invitation and will present to us today in the realm of this SPHN webinar, the OMOP Common Data Model, a model maintained by the global OHDSI open science community and used successfully by many organizations and researchers worldwide. He will discuss the relation between the OMOP and other healthcare data standards as well, such as FHIR or CDISC, and also put this information into the context of the recently published release of the SPHN interoperability framework, leveraging RDF and semantic web standards. I will put you some information about the SPHN interoperability framework later in the chat. And with this, I would like to warmly welcome our speaker of today. Kees van Bochove is founder of the Hyve, a company dedicated to the support and facilitation of open science, open source, open standards, open data in biomedical informatics. Kees has contributed to many open source biomedical software development communities such as the OHDSI, but also I2B2 Transmart, CBioPortal, Radar, GA4GH, and others, and is active in pre-competitive projects such as the IMI EHDEN project, Pioneer, Big Data at Heart, H2O, FAIR Plus, Go FAIR, and many others. He actually has been involved with the FAIR data movement since the initial Lawrence workshop in 2014. Today, Kees's main expertise and engagements are as principal consultant advising pharma companies, but also academic hospitals, as well as patient and health data networks on their FAIR data strategy and advising and leading implementation projects with teams from the Hyve. In this role, he has assisted many companies and academic initiatives, among them also the Swiss Personalized Health Network initiative at its very beginning in 2017, 2018, if I remember correctly. So I'm very grateful to give the floor now to you, Kees. Thanks a lot for taking the time and being with us today. The screen is yours.
Well, thanks for inviting and having me, Katrin. It's a real honor to present to our Swiss friends and everyone that's online today or watching later. So when we discussed the topic of this webinar, you said to me, I've watched some of your previous webinars and you're welcome to go a bit more in depth, which is great, but it's also a bit of a challenge because there's so many disciplines involved in this topic. We have epidemiology, we have molecular biology, we have the computer science aspects. And so going deep into one of them means skipping out on others. So what I've tried to do is to, throughout the presentation, especially become very concrete and give a lot of examples so that hopefully keeps being interesting for everyone that's joining. So I want to start with a bit of introduction. Now, some of you may already be familiar with those concepts, but I think it's good to frame them anyway. Then the main thrust of the presentation today is to really discuss a specific article as an illustration of what OMOP and OHDSI really can do and what they're meant for. And then I'll conclude with some brief remarks about where it fits in the healthcare data space, but I'm also hoping to get some discussion and question on that. All right, so we're talking about open science. And the first question is, how do we even define that? Now, it's hard to get a formal definition, but I always like to use this picture from my alma mater, Utrecht University, which explains that where maybe in the past science was more of an isolated effort, today it's very much part of our society. And there's no other time where we can see this better than probably today with the coronavirus pandemic, because we have scientists producing reports that directly inform government policies that may even go as far as restricting your movements. You have questions about what is the safety of vaccines. And so it's now more important than ever, I would say, to be very clear and transparent in what data do we use when we produce medical evidence. And this is a key to what I will present today. It's also key to the Hyve. So the Hyve as a company, our mission is to enable open science by developing and implementing open source solutions, and also by verifying data. And one question I often get, which I can get out of the way, is how do you make money? Well, we simply provide commercial support, and we do that to many organizations, including pharma companies and hospitals, but also we engage in many pre-competitive projects. And this is really about working together in open source communities like OHDSI, CBioPortal, Open Targets, FHIR, etc., and building solutions that we can reuse in multiple organizations. And here you can see some of the Horizon 2020 and IMI projects that we're engaged in, also active in many partner communities. And as you mentioned, Catherine did some work with SPHN and also with other health data networks to set up the infrastructure. One project that I want to highlight in particular is EHDEN. So the EHDEN project is a 6-year project, which is now approaching the halfway point. And we have in there a number of small companies, also public bodies like NICE from the UK, and a number of pharma companies. And the mission of EHDEN is really to also promote open science, but in the end to enable better health decisions, outcomes, and care. And this probably sounds familiar. I think SPHN has similar goals, and I think many of you today are online will have those goals. But a specific focus of EHDEN has to do with this second E. In the end, the project is all about producing reliable medical evidence. However, that's of course not, and I will provide an example, but that's not the full story. We also have the values of EHDEN, and I want to highlight one of them, and that's community. Actually highlight this in 3 different ways, telling you a little bit more before we dive into the paper and really what OMOP is about. Tell you a bit more about OHDSI, about the community of EHDEN, and also about a new phenomenon, namely study-a-thons. So the community of OMOP OHDSI, OMOP really started more than a decade ago as an experiment in drug surveillance and looking at adverse effects of drugs. So this was the first OMOP experiment. There was also a European OMOP experiment. And so this gave rise to the very first versions of what we now call the OMOP Common Data Model. We'll see what that's all about in a minute. But importantly, this model was also used to set up a community, the OHDSI community, observational healthcare data science and informatics. And we've had a number of symposiums, really great gatherings of a multidisciplinary community, as you can see here. So we have stakeholders from pharmaceutical companies, not surprisingly, and from, for instance, the FDA and EMA, etc. So healthcare agencies, but also from hospitals, from other participants in the health system, people representing primary care or veteran care. And then, of course, we also have the technology providers and the data experts, etc. So if you look at the OHDSI network today, it's about having data in the OMOP Common Data Model. And a conservative estimate is that we already have 500 million distinct patients throughout the world, and making this the largest healthcare data model, to my knowledge, in terms of at least the number of patients represented in it worldwide. Now, something about the EHDEN community. So for us, it's not a nebulous term. It's very concrete because we have many stakeholders, but we focus on building relations with a few of them through programs. And one of the programs we have in EHDEN is our EHDEN Data Partner Program. We have a budget in the project of 17 million euros to help healthcare organizations in Europe to map their data to this healthcare data standard, the OMOP Common Data Model. And we do that through regular data partner calls. We just had our 4th call, and I think we added some data sources from Switzerland in that as well. But overall, we have an estimated 200 million healthcare records represented in these data partners that are now working on mapping their data to OMOP. Another program we have is recognizing that we need more expertise in this field, is to train small and medium enterprises, companies, again, across Europe or anywhere through the Academy, I'll come to that in a minute, can apply to EHDEN, become a certified SME partner. And this means that you've had, as a company, a basic training in the use of OMOP CDM and the OHDSI tools, and that you understand how to do, for instance, the conversion from healthcare data as its primary source to OMOP. And we're also growing this network of companies that have expertise. Finally, I already mentioned it, the EHDEN Academy is one of the tools that we use to do this. So in the Academy, we have a whole bunch of courses that are focused on anything from, how do we install OHDSI tools to how do you generate medical evidence. And actually I want to highlight one course, which is the population level effect estimation course. And you'll see that when we will discuss the paper, this course would provide you with a very good background and understanding also the epidemiological principles behind it and why the OHDSI tools work in this way. EHDEN Academy is free for anyone, even though our programs, because of our European funding, are focused on Europe. Okay, so the last community aspect I want to highlight from EHDEN is study-a-thons. And so let me take you back in time a little bit to spring last year. We had this, I would say, frantic search for medical evidence. You probably remember it. I certainly was reading a lot of papers at the time because we had this new virus and the pandemic. You also had medical claims going all over the news, up to the US president, for instance. And so it became really, really important at that time to have a reliable way to establish medical evidence. And the OHDSI community and also the EHDEN project recognized that. We originally had planned a gathering in Oxford in March, but we repurposed this to be a virtual meeting and decided to sort of project the full force of the OHDSI community to solving a number of important issues and addressing research questions that had arisen in that moment. So we had hundreds of collaborators calling in from across the world. And interestingly, we had about 12 questions. And going a little bit through these questions also allows me to explain one very important aspect of OMOP and OHDSI, and that is, what are the types of questions that OHDSI answers and that you can use? We have three basic categories of questions or use cases, and they are also described in what we call the book of OHDSI. You can see the hyperlink here. And these are characterization, or put very simply, what happened to the patients. Prediction, which is about what will happen to me. Can I predict the future based on what my health history to date? And also about estimating causal effects. Now, just to provide a little bit of more examples here. So in characterization, what you typically do is you have a healthcare population, for instance, can be from a hospital electronic health record system. It can be from a primary care system. It can be from a registry. But you look at the patient and you describe what's happening to them. So you get things like incidence rates, cohort counts, etc. And you can stratify your population and then see how these different strata compare. In this case, it was, of course, very interesting to compare the COVID-19 patients to data we had from earlier years from influenza. So that's really, and treatment pathways is another example of characterization where I'm interested in what are the treatments used in real life for a specific condition. Prediction is all about, can I predict what will happen or what's likely to happen? And one interesting question in COVID was, if you have a patient at the hospital and the condition is stable, but what will happen if you send them home? Can we predict if they will need to come back anyway and require further hospitalization based on the data we have so far from other COVID patients? So this is one of the models that that was created and also published subsequently. The third category is to look at estimation. Now, this can be about, for instance, the safety of a drug. This would be the example of hydroxychloroquine, where this was a drug used in rheumatoid arthritis, but suddenly became very well known, and the safety profile of the drug hadn't been studied in like the general population. So it became important to do that, and indeed, OHDSI was able to do it quickly. But another question, and this is where I will actually focus on, has to do with ACE inhibitors. All of these questions produced preprints, and those preprints eventually also led to papers that are peer-reviewed. Some of them are in preparation, some of them have follow-up efforts. You can see the full list here on the OHDSI website. And this is just a selection of a few papers. And as said, I'll focus on the second one there. What I want you to take away from this introduction section is that OHDSI, and also specifically EHDEN, is building a federated health data network. EHDEN is the European variant of it. And we focus on generating medical evidence using open science principles. Let's see it in action. So if we transport ourselves back to March 2020, what do we know at this time about the renin-angiotensin system? Now, this is a system. You can see here a pathway diagram, which we can have from system biology papers. I took one from this paper here. And you can see that in this system, a very complex and important system, one of its functions is about regulating blood pressure. We have actors like the ACE protein, and the ACE inhibitors are drugs that inhibit this. So let's go and look at a few facts. We knew from earlier publications coming out from China, for instance, that hypertensive patients were overrepresented in COVID-19 hospitalization cohorts, meaning that those patients tend to have a more severe progression of the disease. We also know from literature, from clinical practice, that many of those hypertensive are on what's called RAS blockers. So these are ACE inhibitors, but also medicines that block the angiotensin receptor. That's the AT1 you see here in the pathway diagram. And it was hypothesized that if you block this, let's say, red part of the system, then this would lead to a further activation of other routes in this pathway. And indeed, it was also shown, and now the picture is more nuanced, but at the time, the main hypothesis was that those RAS blockers increased expression of ACE2. However, we also know that the ACE2 receptor happens to be the way for the coronavirus to attach itself to the cell to gain entrance to the replication mechanisms like the ribosome, and really in the end reproduce itself. So, if you put all this together, you start thinking, what if this is not a coincidence? What if through all these patients that are on RAS blockers, we are inadvertently rolling out the red carpet for the virus, and by sort of pushing the cells to express more ACE2, making it easier for the virus to enter the cells and to replicate. And indeed, this was a hypothesis that was picked up by many. Here's just one example from Switzerland, Michael Roth, that published this, let's say, letter or opinion article. And you can see here that he says, these data suggest that ACE2 expression is increased in diabetes, and treatment with ACE inhibitors and ARBs increases ACE2 expression. Consequently, the increased expression of ACE2 would facilitate infection with COVID-19. So now this was a really important question. Is this true? How do we go about checking if this is indeed the case? Well, ideally, what you would do is you would perform a randomized clinical trial where you give a portion of the patients RAS blockers, and you give a similar population of patients another drug. But of course, in the setting of the pandemic and the speed that we wanted answers in, and for all other reasons, this was not a practical approach. And also this of not what OHDSI about. OHDSI is about, and the OMOP Common Data Model is about, can we see this in existing data that is, for instance, routinely produced in the healthcare system? So naively, one comparison that you could do is to take COVID-19 patients that are on RAS blockers and look at their disease progression, and then compare that with COVID-19 patients that are not taking RAS blockers. However, if you do that, you introduce quite a bit of bias because, of course, first of all, if you're on a RAS blocker, you likely have hypertension. So you may just be looking at the effect of hypertension rather than of this specific drug. And the hypothesis that we had was based on the molecular mechanism of the drug. So a better idea would be to take COVID-19 patients that are on RAS blockers and then compare the outcomes they have with patients that are on sort of alternative, comparable hypertensive drugs. Now, this doesn't fully mimic maybe the 50% randomization, but you can use statistical methods to make sure that we get as close as possible to this. How does that work? Well, one method that's used a lot in OHDSI is propensity score matching. What is the propensity score? It tells me for a patient, if the patient a priori, just based on the medical history, what I know about the patient, is more likely to get the what we call the target drug. So in this case, it would be an ACE inhibitor or an angiotensin receptor blocker, versus the comparator drug, the drug that I'm comparing with. And in the paper, we take classes like the calcium channel blockers and thiazide diuretics, which are comparable drugs. And you can see here these nice plots. In fact, you can try this out for yourself if you go to this URL, because OMOP and OHDSI is all about publishing medical evidence openly, and also not just for specific research question, but for general usage. And the LEGEND study does a fairly broad exploration of hypertension and the drugs used in hypertension. And what I can see here is that if I take specifically ACE inhibitors and these type of calcium channel blockers, that yes, mostly these, let's say blue and red overlap. So, but there's also a significant number of patients here on the left, for instance, that based on their medical history are much more likely to get the blue drug. And here there are patients that are much more likely to get the red drug, the ACE inhibitors. So we can't just do a simple comparison. However, we can create, fit a propensity score model. And then what we can do is we can do match. So we take a patient that's on an ACE inhibitor and we can find a patient that has the same score, so is sort of equally likely to get that ACE inhibitor, but happens to have gotten a calcium channel blocker. And by using this pair, I make sure that I adjust for this bias that exists. And indeed, if you look at the supplementary material of the paper, you will find loads of all these plots. So this is for every combinations of drugs that we looked at, for every data source. You can see that, so I mentioned matching, but another method is stratification, where instead of pairing up individual patients, you would look at the, let's say, strata. So could be age strata or other types of strata where I compare patients that way, and that way I can use a little bit more of the data. So this is all about correcting for observable confounding. So that's things that we can see and reason from the data. However, there are still also confounders that we do not have in the data, but that may have an effect. Can we control for these too? And yes, it's possible to some extent to correct for unknown confounding by using negative controls, so things that we know are not associated with the disease. It would go a bit too far to go into detail now about that here, but I can highly recommend you to take the course on the EHDEN Academy or take some read the book of Odyssey, for instance, to look at this further. Now, for the study in question, you don't need to go to the supplementary material. We also have conveniently created an app where you can see all these plots, and you can see that the same one, that this is like a screenshot I took from the PDF of supplementary material, but you can see the same plot here in the interactive application. And this gives me then the propensity score. What is really cool is I can also look at the propensity score model. And now I can talk to a clinician about this. So, the data tells me that if people have a transplanted kidney, they are more likely to have one of the drugs. So what is that about? And you can also Google it. So I'll leave that as an exercise. But in principle, there should be ideally explanations for why the model found these. Okay, so that is the statistical part. However, there's more to OHDSI and OMOP than just the statistical methods choice, because a very important other aspect is the definition of cohorts or phenotypes. And we have the Atlas application that we can use if you have data in OMOP. And it's interesting to look at the cohort definitions that we used in this study. So we looked at drug exposures of, for instance, ACE inhibitors would be one, an ACE inhibitor monotherapy. Now, normally you would look over a long period of time, but here, because we have the coronavirus pandemic, we knew that there were likely to be almost no cases, let's say in January 2020. So we are looking here in a 2-month period for our people on ACE inhibitors. And we have a number of other criteria as well. And this really makes it a cohort. So as we like to say in OHDSI, a code set, a set of codes is not a cohort. We really need to define what is the entry criteria, what is the exit criteria. And as you can see, the monotherapy aspect here is listed by no exposure to other antihypertensive. And also we have the hypertension diagnosis. So this is just to make sure that we are already selecting hypertensive patients. Just to show a few features of the system. So if I hover over the drug exposure, I can see what is actually included. So these are ACE inhibitor drugs. And if I then go to my what's called concept sets in OHDSI, I can see that there is actually 10 drugs included here. But I don't know if you can read this, but the number of included concept is more than 20,000. And the reason for this is that even though we select this drug, this leads to many specific codes in different formulations of the drugs that could be recorded in the primary healthcare data sources. And therefore, we have to include all these derived concepts. And the OMOP OHDSI tooling makes extensive use of the vocabularies or ontologies that are important, and especially the hierarchy in those to do that automatically for you. So I just click the sentence here, and then I have all these concepts included. Now, also in the inclusion criteria, the time aspect is important. So I have to specify what the inclusion criteria are from the start date, and then whether I include, let's say, everything or up until a specific, up until the exit of the cohort. Okay, so that is the definition of the cohorts. And now we have a number of important aspects of the study design. We have the exposure, which gives us our target and comparator cohorts. So again, target would be something like ACE inhibitors and angiotensin receptor blockers. The comparators would be similar drugs. We have the method, so adjusting by using propensity scores, etc. And then in the end, computing the hazard ratios. But another important question is, what are the outcomes that we want to look at? We're interested in whether those class of medicines either, let's say, increase the risk of COVID or make your COVID progression worse. And we do not have that literally in the data, but we have proxies for them. So one proxy would be COVID-19 diagnosis. And actually creating the cohort for COVID-19 was not that easy either. Of course, there's a SNOMED CT code, but there's also a LOIN code. So you could have a diagnosis record, but you could also have a record of a positive test. And then because at the time there were, let's say it was a new disease and not everything was immediately standardized in healthcare data collections, throughout the dozens of healthcare data sources we used in the study-a-thon, we also had specific other codes that were used. Okay, so that gives me COVID-19 diagnosis. But another important thing would be to look at hospital admission with COVID. And recognizing that maybe some of those cases weren't recorded, we can also look at pneumonia. Now that, of course, may then also include influenza patients, but this is why we look at multiple outcomes and then try to interpret the results we see as best as possible. Finally, another important mix of study design in this kind of observational studies is your time at risk. And here that's defined as the day 0 is the last prescription record we have, and then until the end of data availability, or like if people get one of the outcomes, etc. You can read the details in the paper. All right, so this is what we need to do generally when we do a study, but it's not the only story, because once we have the cohorts defined and we have the method defined, we create a study package, and then we also have to the logistics of the execution. And this is where the Federated Health Data Network nature of OMOP OHDSI comes about. So we send this package to multiple sources, and in the end, by combining these results, we then get the full interactive application that I just showed, but also we get the estimations that we're looking for. And here's the end result. The cardiologists and the doctors, they can, let's say, breathe with relief here, because it turns out that if we look at hazard ratio estimates that we eventually came up with, a lot of them are around 1, meaning that the data supports the hypothesis that there isn't really a difference in those outcomes if we compare against these drug classes. Neither in monotherapy nor in combination use, because let's say if you are, the important point is to compare between ACE inhibitors and angiotensin receptor blockers versus other classes. So if you take a combination of those drugs, you can still include these people in the comparison. Gives us a little bit more data, so some tighter intervals, but in the end provides the same picture. Okay, so that was the result of this paper. Now we can generalize this also, and I would recommend, if you're interested, to look at this paper from Matai and Shumi, where they looked at depression treatments, so psychiatric drugs, and then at the literature, so what is really published in literature. And this is really interesting. So if you look at all observational literature and you look at the spread of estimates that are being published, you see a real bias, let's say, against p-value, but you also see that you have very few confidence intervals that include 1. So, no difference basically. And then you see the same if you focus only on literature with depression treatments. However, if you just publish all the estimates that you can generate, as the OHDSI community has done with the legends, with this study, you see that you get very different distributions of the the estimates. So meaning that there is really a bias in this literature. Now you can speculate as to where that bias comes from, but I think that the spirit of the OHDSI community is important to publish all of this as open science, so that that's available as reference information when you make a medical decision. And we do not just cherry pick the results that we're interested in. So hopefully in the second part of the presentation, I have convinced you that medical evidence generation is really changing now from paradigm. So instead of, let's say, one study where we look in one database with one hypothesis and we compute one or a few estimates, we do a large scale study where we have multiple databases across the world and we generate a range of estimates. For instance, all estimates that have to do with a particular disease or a class of drugs. And if you're interested in learning more about this, there's the EHDEN Academy, but there's also the book of OHDSI and some of the papers that I highlight here. Now, the last few minutes, I'd like to focus on what does this all mean in terms of the role of OMOP OHDSI in the larger biomedical health data standard space. And we've also published an EHDEN deliverable on this. It's called Roadmap for Interoperability Solutions, but it discusses let's say a little bit more in detail some of the similarities between these models. But the key really is that you need to ask yourself what the use case is. And in my opinion, all of these standards that we've worked with in the Hyve have their own place and their own use case, and they can perfectly coexist with each other. If you think about that a bit further, the blog from Graham Grieve, which is one of, let's say, the fathers of FHIR, very active in HL7 for a long time, distinguishes between three legs of the healthcare data standardization process. So there's development of base standards, and these are very important, let's say, commons that we all rely on. So for instance, SNOMED-CT, I don't have to explain it here because SPHN webinar series had a previous episode that did an excellent job of explaining SNOMED-CT and LOINC, etc. But these are really base standards. And ATC for drug classes, UCOM for unit of measurements, ICD classification of diseases, HPO for human phenotypes, etc., etc. So these are, let's say, the base layer that try to model a particular domain. But then the second step is that we also can profile these standards for a particular community. And I think the SPHN interoperability framework is an example of that. You could look at other examples like eHealth Swiss is how do we exchange healthcare data in Switzerland electronically. FHIR profiles are an example of this, where you create specific FHIR profiles for a community, like the MedMij FHIR profiles in the Netherlands, which are for healthcare data exchange between general practitioners and patients, so that patients can see their own data. OMOP, I think I would have convinced you that is also meant for a particular use case and community. And the third leg of the healthcare data standardization is to really drive this into the market, because a standard is more interesting the more people use it. And so adoption is very key. And I almost like to think that in the OHDSI community, the goal was always, let's say, producing medical evidence. And then everything that's done in OMOP is sort of reasoning back from there. So, okay, we need some healthcare data standardization. We need to develop these statistical methods. We need to have cohort browsers. We need to have tools like Atlas where we can create cohorts, etc. Okay, so how does this play into OMOP and OHDSI? The OMOP Common Data Model, in case you don't know about it, I think I've explained the function, but just how it technically works, it has a number of tables, a number of SQL tables. And there's a table, for instance, for the drug exposures. There's a table for the device exposures. There's a table for the conditions. So this would typically use SNOMED-CT codes. If your data is not in SNOMED-CT, then we create a mapping from the source vocabulary to SNOMED-CT. We have measurements which are in LOINC, etc., etc. Now, these mappings that I just mentioned, you can also find these in Athena, one of the OHDSI tools. And I think there is more than 140 vocabularies. So these are source vocabularies, which could be as specific as the Swedish variant of SNOMED CT, or ICD, let's say ICD-10 SE. But also research ontologies, anything really that's used by the healthcare data sources. And we map this to this model. This allows us to do the standardized analytics. So the statistical tools that I showed earlier are coded in R, some in C, and they are gathered up in a package called HADES. Now, just to drive the point home that you need to look at use cases, I took a little bit I took the data summary of the CPRD, which is a UK primary care healthcare database, clinical practice research data link. And if you would project it onto Switzerland, you would get something like, and this is very rough estimations, I may be off here, but the order of magnitude overall, I think, is correct. You would have, more than 10 million patients, because you're looking back in history. You have billions of medication records. You have billions of observation records, and so in the end, you have many billions, probably getting to the trillions of triples just to represent the raw data, and that's not even counting the vocabulary. So obviously, let's say trying to put all this in a triple store is not something that you would want to try. And so that gives us then a nice segue to, this is my last slide here, to compare some of these healthcare data standards. Obviously, I don't do them justice. I can't do that in a few minutes, but still, I wanted to put something on the screen. And I think if you look at the difference between them, an important axis is the flexibility. So let's say in RDF, you're extremely flexible. But of course, you still have to pick out the URIs that you use, the ontologies that you import, etc. With FHIR, you also have very high flexibility. FHIR is meant for this. You can create profiles for the base resources of FHIR, and indeed many do so, as I have lined out some examples. CDISC, on the other hand, is a bit more prescribed, because there it's really about submitting data to regulatory agencies, and there's a format for that. So adverse event and CDISC have, let's say, a slightly different meaning, concomitant medications domain, than what you would have in OMOP in terms of the same data types. I2B2, for instance, also is quite flexible. You can define your own ontology in I2B2. Again, the EHDEN deliverable goes more into detail about it if you're interested in. But OMOP doesn't allow me a lot of flexibility. It's really a one-size-fits-all approach, and that is very deliberate, because what we want to do is we want to be able to do standardized analysis across the globe. And to do that, we need to really make sure that the data is syntactically and semantically similar and comparable. A framework that I sometimes like to use to compare data standards is a very simple one. Maybe you know the 8 W's of project management, but this is just to ask, where does this data come from? Where is it generated? What is the original context? So for FHIR, for instance, typically those are healthcare systems that expose FHIR endpoints for interoperability purposes. In CDISC, I would be gathering my clinical trial data at the sites. I2B2 is typically used in the context of an academic hospital, and OMOP is used in many different types of databases. Then there's the question of what do we have? What is the type of data that's in there? And OMOP would be great for, as I showed, trying to estimate the risk of taking ACE inhibitors, but it's never going to tell you that SARS-CoV-2 binds to the ACE2 receptor. You need a different type of database for that. Okay, so I think I'll stop here because I want to have enough time for questions. But I think it's clear that if you look at where does the data sit, what is the type of data, and what is the use case for it, all of these healthcare data standards have their own focus, and they can coexist, and you have to think about what tool to use for your question. All right, let me then end by inviting you, if you want to become part of OMOP Odyssey, to join the journey, and specifically in EHDEN. If you have health data, you can become a partner. If you are a company, you become a certified SME. If you're a research, you can join our network studies. If you're a pharma company, you can adopt the OMOP CDM. And if you want to learn more in general, take a course in the EHDEN Academy. Thank you so much.